To introduce Dr. Percival McCormack, who is a native of Ireland and received his MD in 1978 from Trinity College in Dublin, and prior to that obtained a PhD in nuclear physics. From 1979 to 2000, he was a U.S. Navy doctor and flight surgeon, and for 6 years was assigned to NASA as a chief of operational medicine. He also served for 6 years as Chief of Environmental Medicine at the Navy Medical Research Institute. In 2000, he took his present post at the University of Illinois at Chicago. He works on imaging techniques such as ultrasound and MRI, and also in nanofluids, which is a topic of interest for him, and he's writing a textbook on the subject, the vortex, molecular spin, and nanovorticity. One item that's not mentioned in Dr. Percival McCormack's bio is that he was on the faculty at Thayer for about 3 years, sometime in 1970, right? So welcome, Dr. Percival McCormack.
Thank you very much. Do I use this microphone?
Yes, you want to turn it on?
Just press
the button.
Here, I think.
It will be on, yes.
There it is.
Yes.
That's good. Now, can you hear me? Is the loudspeaker working? Okay, good. So thank you very much. I'm very glad to be back here at the Thayer School again, and many happy memories of being here as a professor many years ago. I've talked to some of you today already about the radiation problem for human space exploration, and so this it's kind of maybe too concentrated for many of you. It's going to definitely concentrate on that aspect of radiation biology, and but that's what I've been involved with and still am. I just came back from a NASA meeting in Houston. They had a meeting on space radiation workshop at Houston, and I've just come back from there. So I still keep close connections with NASA. I very much believe in the human space exploration of space by humans, but at this stage we have run into a little hiatus, and that may take some years to resolve, but I know it's going to go ahead in the future. So I'm going to talk about a major problem. Because for flights beyond the Earth's magnetic field, and I mentioned that it is the magnetic field that's the critical factor relative to the Moon or Mars. At go to high altitude, the galactic cosmic radiation predominates, and this is comprised of high energy particles from protons to iron ions. Now, the reason they're unique is that they have extremely high energies, billions of electron volts, and they create damage in biomolecules that can result in long-term changes like genetic mutation and cancer induction. And these are the these are the biological changes that determine the major risk of long-term space travel, something that humans have lived with here on Earth since their creation, but we're protected, as we see, by nature's ingenuity. Now, the galactic cosmic ray radiation field has a broad maximum near 200 MeV per nucleon, followed by a continuous decrease towards higher energies. And transport calculations have been done because obviously to protect, the first line of defense is to use shielding materials. But transport calculations for the GCR spectrum in various shielding materials like aluminum show that after a small decrease in the first thin shielding layers, thicker absorbers do not produce a net reduction of the biological effect that one might expect from the increasing mass of the shielding material. So shielding is not very effective as a countermeasure against galactic cosmic rays. It is against solar radiation, don't get me wrong, but not against galactic cosmic rays. So reduction, you could try shielding, but as I said, the decrease in the amount of low energy particles is almost compensated by the increase in nuclear fragmentation. The showers come out from where these particles hit. And to protect the astronauts from the damaging biological effects of GCR, what I'm talking about today is a proposal to develop an alternative method to shielding. Shielding doesn't work, really. It works maybe 10 or 20%, but not very effectively, and so we need an alternative method. Now, what's the injury? The signal injury from GCR exposure is induction of DNA double-strand breaks. And in mammalian cells, it's found that the first line of defense against these double-strand breaks is the direct ligation of the broken DNA ends via the non-homologous end joining pathway, and I use NHEP for a short version of that. So that's been a sort of a first line of defense, non-homologous end joining. The join, the end, literally where the break occurs, the ends are joined back together. And it is proposed to develop, and I propose this to NASA, a research program to strengthen this first line of defense and extend the stay time of astronauts on the Moon or Mars. I've already done calculations on this, and as there's no magnetic field around the Moon or Mars, they don't get that protection that we have here. And so whereas we can live 75, 85, whatever, going up years here on Earth, on the Moon, I've published papers on this. I've predicted that we could only stay 2 years on the Moon or 2 years on Mars. And if you're going to Mars, 6 months there and 6 months back, you've got 1 year on Mars. So it's likely, however, that nanotherapeutics, a rapidly expanding area of medical research and development, will play a major role in this strategy, because it's quite complex, the whole thing. Now, what is the space environment radiation risk? Astronauts are exposed to protons, high-energy heavy ions, or HCE, with high charge and high energy, plus secondary radiation, which are mainly neutrons. The energy spectrum of the GCR I mentioned before is maximum near 1,000 MeV per nucleon, so penetrating that shielding can only partly decrease the dose received by the crew. Also, thick shielding has problems for launch systems due to the extra mass and can only reduce the effective GCR dose by about 25% using aluminum and about 35% using polyethylene. So, except for solar protons, shielding cannot be a solution for the problem of radiation exposure in space. Now, biophysical models show that energy deposition by high LET radiation produce different DNA lesions, including complex DNA double-strand breaks. The number of single-strand breaks and double-strand breaks varies little with radiation type, but for high LET radiation, much DNA damage is complex and consists of mixtures of single-strand breaks and double-strand breaks and occurs within localized regions of DNA. And repair of these DSBs, as double-strand breaks occurs through direct end joining and homologous recombination. Multiple double-strand breaks result in little repair and cell death. The risk of cancer increases. So does the risk of genetic changes. Now, the maximum level, the kind of guideline that has been adopted for radiation risk has been and will be, as far as we can see into the future, 3% fatal risk. That is, if you get up to 3% fatal increase in risk of cancer, mortality from cancer, then that's the maximum. Something has to be done to try and avoid that or step out of that environment or whatever. And this thing I've referred to, it's called non-homologous end joining, NHej. And in this mechanism, the broken ends are brought together and rejoined in the absence of long tracks of sequence homology. And this type of repair is found to occur in organisms ranging from bacteria to mammals, indicating it has been concerned during evolution. Nature has developed this technique, which is amazingly ingenious. And in conceptual models of the NHej mechanism, which this diagram shows, the ends are simply brought together and then joined, as you see here in the last two straight lines. First were the undamaged DNA, and then the end two lines show the repaired DNA. A problem, however, may occur that if the cell incurs more than one double-strand break, then previously unlinked DNA molecules may be joined, resulting in gross chromosomal rearrangement. But in mammalian cells, it's found the correct ends are usually rejoined by NHej. And the binding, as we've seen in the last picture, of specific protein factors to the broken ends is the first step in NHej. The key step in NHej, of course, is the physical juxtaposition of the DNA ends. If they can be directly ligated, then the repair event is quite simply repaired, in that it requires the action of a DNA ligase for completion. But most DSBs generated by exposure to DNA damaging agents do not have ligatable termini, and so they must be processed prior to ligation. That complicates the procedure, the repair procedure. This implies that partition of enzymes that remove lesions such as phosphoglycolates from the DSB terminal NHej. Then after end bridging, there appears to be an alignment step in which the DNA sequences close to the ends are compared and then aligned, and that was all shown on that first figure. So that's the simple situation. And now the biochemical properties of the NHej factors. There are many factors. Genetic and biochemical studies have identified certain complexes. It's not that simple as just that first diagram I showed. Certain genetic and biochemical studies have identified what's called the Ku complex, which is a heterodimer of Ku70. And there's Ku86, also known as Ku80, as the major DNA binding factor. And this is shown in figure 2. I think this, the writing at the bottom of the figure is too fine for you to read, but I'll just read it off here. The bacterial and eukaryotic Ku proteins share a similar central core domain. And however, the eukaryotic proteins contain additional nitrogen and carbon terminal domains. And that's presumably to support interactions with various protein partners found in higher organisms. So can't go into all the fine details of this, but I give references, and I leave the paper here, and the references are there. And if one wants to learn the procedure step by step, then one has to go back to the original papers. The crystal structure of Ku, complex with DNA, revealed that the heterodimer, which they form, Ku and DNA, has a toroidal shape with a central hole big enough to accommodate duplex DNA, and the reference is given there. The interior of the asymmetrical ring is lined with positively charged amino acids, allowing for sequence-independent interaction with the negatively charged phosphodiester backbone of duplex DNA. And the activity of Ku in end joining, and that's the significant unit that's involved in end joining, is dependent on a material called anisotol 6 phosphate, IP6, which binds to the Ku complex. And a common emerging theme for different DNA repair pathways is the role of the protein-protein interactions in the assembly of multi-protein repair complexes, and this is shown in the next slide. And this is a model for mammalian NHej only. And so in mammalian cells, induction of a double-strand break, which is shown in A in this figure. And this is followed by the recruitment of the Ku heterodimer. That's very, very important. It's in B. And this Ku, that's a vital step. And the third step then involves recruitment of DNA, PKCs, and Artemis. It's another material which is vital to this procedure. So you can see nature is amazing the way it brings in these necessary compounds, which then are involved in the step-by-step repair of this damaged DNA, resulting down right at the last stage, G shows, in ligation again, and you get, hopefully, and as it turns out, truthfully, a nicely repaired DNA double strand. By virtue of its localization to the double-strand breaks at an early step in the non-homologous end joining, DNA PK is well suited to recruit other DNA repair factors to assist in the repair and processing of the lesion. The core NHej factors are also required for completion of the VDJ recombination initiated by the site-specific RAG1, RAG2 endonuclease. And following the alignment and nucleolytic processing of the broken DNA ends, there are usually small gaps that have to be filled prior to DNA joining. The mammalian POL X family members in the last figure, 3F, POL-U and POL-lambda have been implicated in the gap-filling reactions associated with NHej, and the references are given there. And I said, really, to follow this in detail, you've got to read work from the references. And I leave the paper here. In mammalian cells, remember, the first line of defense against double-strand breaks is the direct ligation of broken DNA ends via the non-homologous end joining pathway. But repair can also be accomplished by homologous recombination, and that's shown in the next figure. And just during HR, a homologous sequence, usually a sister chromatid, is used as a template to receive the genetic information to repair the damage. In contrast, NHej rejoins the two DNA DSPs directly without requiring sequence homology between the DNA ends. Let's have a look at this figure 4, which is the next one. And this is now a model for one specific type of NHej based on S. cerevisiaceae. It's following the induction of a DSP, which is shown right at the top. You see the double-strand break in A. And following that induction, the initial step in this repair is end binding by what's called YKU, and that's in B. YKU is the end binding mechanism. The MRE and D50 over XRS2 is actually a complex associated with DNA-bound KU to form an end-bridging complex that's in C. That's what's diagrammed in C. The NHej-specific DNA ligase, DN4, which is shown here in D, and its associated factor are recruited to the break site, and then promote processing and gap filling by recruiting the RAD27 endonuclease. You can see the RAD27 endonuclease and the NHej polymerase POL4. That's C. And then once ligatable terms have been generated, DNA 14 repairs the lesion, and you get again at the end here everything ligated and the damage repaired. Homologous recombination also provides a pathway by which stalled or broken replication forks can be reestablished, thereby allowing cell survival. And in this paper, I don't deal with that specifically. Now, in the next section, I just want to conceive of what would be sort of something one could aim at in trying to understand this repair mechanism which nature has developed, but also to be able to design a repair system de novo. And I call that the DNA repair nanomachine. This is the NHej reaction for a DSP repair. The cell nucleus is full of nanometer scale structures associated with functions such as RNA transcription, RNA processing, DNA replication, and DNA repair. These are not enclosed in individual lipid membranes and are self-organizing nanomachines. And this DNA repair goes on continually in human cells, being a response not only to radiation, but to damage from environmental toxins and endogenous oxidative species. The human genome encodes about 150 genes for DNA repair proteins, quite a formidable defense. These proteins organize to form about 12 different types of repair nanomachines, each specializing in a different type of damage. Double-strand breaks and maintenance of genome integrity. Ionization causes DNA DSBs leading to potent biological effects. Now, the DSBs lead to chromosome breaks and to production of acentric chromosomal fragments and so on. Even an unrepaired DSB may result in significant loss of information during cell division and to cell death. Passage of high-energy ionizing radiation through tissue generates reactive chemical species distributed inhomogeneously along nanometer-scale tracks. When a radiation track intersects the DNA helix, both strands are damaged simultaneously, frequently with the formation of a DSB. DSB repair starts once the damage is detected, with the rapid component having a half-life of only 7 to 14 minutes and a slow component of 60 to 90 minutes. And two different types of nanomachines act in the repair of DSBs. As mentioned above, only the non-homologous end joining will be considered here, in which DNA ligase directly rejoins broken DNA ends that have little homology and often lead a small region of altered sequence at the break site. And NHej appears to be the main path at clinically relevant radiation doses. Now we look at something to do with the structure, function, and assembly of the NHej nanomachine. NHej occurs within visible subnuclear structures called repair foci, or ionizing radiation-induced foci. Such structures are defined by the presence of a domain of modified chromatin enriched by a phosphorylated histone variant gamma H2AX, which can be visualized with anti-YH at AX antibodies. The foci appear minutes after irradiation and vanish in 60 to 90 minutes. The number of foci approximates the number of DSBs expected for a given dose of radiation. And Figure 5 shows human melanoma cells irradiated with 0.1 gray of ionizing radiation, which is predicted to generate about 3 double-strand breaks per diploid human genome. 3 to 4 foci are seen in these cells 30 minutes after radiation, and after 90 minutes, most of the foci have disappeared. And this is just a diagram and some pictures of what's called gamma H2AX foci. In this SKMEL28, which is the, let's see which one is that, scale 28, that's melanoma cells were co-injected with a reporter plasmid, and either SCFV or control SCFV were also injected. Cells were irradiated at 1.5, you can see that 1.5 gray, to induce approximately 45 DSBs per genome. Control cells were mock-irradiated with 0. You see there the 0 gray in the center of the diagram. Cells were fixed at 30 to 90 minutes after treatment and stained with anti-gamma H2AX antibody panels, and these are the panels that are called A to G. And that's the gamma H2AX antibody. These are antibodies to green fluorescent protein GFP, or DAP1. That's another one you see here in the third line, as described in the reference 35. And GFP is a marker that indicates which cells were successfully microinjected. DAP1 stains DNA and reveals nuclei of all cells in the field. And that's the last line in that diagram. The DAP1 stains DNA and reveals the nuclei of all cells in the field. And the internal structure of the repair foci is still unknown, but one can deduce some facts based on genetic, biological, and cytological studies. From immunofluorescence microscopy, repair foci appear initially as diffraction-limited point sources, which suggests that their maximum dimension is less than 250 nanometers, seen in Figure 5. The domain of gamma H2AX containing chromatin extends about 1 megabase pair on either side of the break. A DNA of this region of this size has a boundary length of about 70,000 nanometers, several hundred times the maximum estimated diameter of repair foci. Cytologic characterization of repair is one approaching to understanding NHej. And genetic and biochemical studies provide another one, which focus on the role of enzymes and other DNA-binding proteins. And figure 5c illustrates the present understanding of the core NHej complex based on these studies. Two halves of the broken DNA are shown as double helices entering from opposite directions, and protein DNA contacts detected by photo cross-linking are shown as circles on the DNA helix. 5 gene products bind directly to broken DNA ends and have a direct enzymatic role in NHej. Ku protein, a heterodimer of 70 kilodaltons, Ku70, and 80 kDNA or Ku80 subunits binds tightly to broken DNA ends and translocates inwardly after binding and recruits the DNA-dependent protein kinase catalytic subunit DNA PKCs. Of the 5 gene products which make up the core enzymatic machinery for NHej, only a phosphorylated form of DNA PKCs has been shown to be present in repair foci by immunofluorescence. Now, the purpose of what's being proposed is to modify the function of these DNA nanomachines to increase the high-energy radiation damage repair capacity. You can see it's a complex system, and it will be quite a challenge to see if one can improve this repair capacity. And of course, modification, what's the objective, is to increase the stay time of astronauts on the Moon and Mars. It is known that single chain, and one thing that has been observed, might be a line to follow, is that single chain antibodies, SCFV for single chain fragment variable, so it's SCFV, can modify nanomachine function. It is suggested that there may be forms of this SCFV that increase the rate of NHej, so it's worth maybe investigating this SCFV. And SCFV 18-2, for example, has been shown to interact with NHej, and in fact, in this case, something one doesn't want, it inhibits it. So that's one you don't want. So there's a lot of investigation that has to go on. This is another figure showing the mechanism of action of SCFV 18-2, the mapping and mechanism. The diagram shows location of fragments and peptides used for epitope mapping relative to kinase catalytic domain. And for details and supporting data, the reference is given. And so if we go to B, then hypothetical mechanism of the action of SCV 18-2, this is the one that blocks the NHej, and so it wouldn't be the one that one would choose, but it's an example of the complexity and the mechanism of action of these different proteins. According to a structural model, SCV 18-2 lies in an extended arm connecting a large DNA binding PAM region to a structural model built on single particle reconstruction. The SCV 18-2 lies in an extended arm, which is shown in the lower part of the diagram, connecting a large DNA binding PAM region to a structural model built on single particle reconstruction. The arm domain undergoes a large conformational change, a bond binding, and we propose that the binding of SCV 18-2 to the extended arm blocks the conformational change and inhibits the DNA binding, resulting in inhibition of the NHej pathway, which is the one, of course, that's not going to be promising, but still one has to understand the mechanisms at work. They're complicated, and they need a lot of investigation. There is evidence for interrelation of transcription and DSB repair that's been noted, reference 22. Nuclear extracts from cells lacking Ku protein, for example, show a characteristic defect in transcriptional reinitiation, and certain Ku mutants have a dominant negative effect on transcription. Ku can be chemically cross-linked in situ to regions actively elongating chromatin. Indeed, for unclear reasons, the loss of Ku function human somatic cells leads quickly in a few days to loss of cell viability. It has been proposed that NHej nanomachines may assemble near or on the active surface of active chromatin hubs that serve as the main site of pre-messenger RNA production in the cell. And the ability of these hubs to organize chromatins into loops may stabilize pairing of broken DNA as the repair machine assembles. And so now I come to some concluding remarks. As strategy for increasing the rate of repair of human DNA damage, DSB mainly, by high-energy galactic cosmic rays, it is proposed that NASA launch a long-term research program to investigate the rate of NHej and how to increase initially by use of single-chain antibodies such as SCFV. And the objective is to increase the human stay time on the Moon and Mars by at least a factor of 2. And then I have a list of the references, and I said to really go through those steps in detail, you really have to look at the references and go through the diagrams very carefully. But the references are here, and I'm going to leave the paper here in the department, and those who are interested can get the copies of the paper and then look up the references. And for those who are interested, it's a very specialized, complex area, but it's definitely a line of attack on trying to improve the repair mechanism for human radiation damage by space radiation. And so I think these are further references. And so let's see what time. I think, is my time now up, I think? Yeah. Yeah, so my time is up.
We're opening the floor for questions.
McCormack, isn't a lot of the galactic cosmic radiation damage doesn't actually kill cells rather than cause DNA damage?
But how does it kill them?
Just by depositing energy in the cell. I mean, it stops in the tissue.
Yeah, well, I'm specifically talking, of course, in this about DNA damage. And the common damage from galactic cosmic rays is the double-strand breaks. Now, that's a small part of a wide range of other possible damages, but which one can go into? I was talking specifically of one type. And then some of the initial ideas in this nanomachine repair mechanism. And you see, you get, even in the lecture, I haven't gone into clearly into all the details of the steps, but you can see it's a complex system. But complex, it's amazing that nature, through the millennium, has identified these proteins and electrolytes and other chemicals which can actually assist in the NHej process, the repair process. And we've got to sort of, I think that's a good line to follow, because if we start in the dark, it might be a long time before we hit on something like, you see one electrolyte or one protein there that actually suppresses the repair. So, you know, we could go through a lot of blind alleys before getting anywhere. I think the ones that succeed in nature, have succeeded and are succeeding in nature, it'd be good if we really got into the details of those and then try to improve on them. It'll be a long, tedious process, no question. Yes?
So, Dr. McCormack, thank you. So, you're talking about increasing the rate of non-homologous end joining, but wouldn't you want to look at homologous repair of DNA, even though it's strand breaks? You talk about mechanism of homologous repair. Is that way you have less chance of misrepair, or did I misunderstand what you were talking about? Because with non-homologous end joining, right, you just have
non-homologous, yeah.
And they get put together whatever way, is possible. So if you have multiple broken strands around, you might get, chromosomal fusion or...
Well, it's just the first step. The NHej is the first step that it looks like could be promising. But homologous repair is also a possibility. Hasn't obviously been investigated as much as the non-homologous, or as easy to understand. But so those are the initial two. I mean, there's probably other approaches, but I just want to get the program started, and I think we need, told NASA, probably 6 or 7 research units centered here in the US or worldwide who will start working on this program in an organized fashion so they don't overlap, but they supplement one another. But to make progress during the next, this isn't needed tomorrow or next year, because that won't happen. But we don't need it that early. But we do down the road when we start the manned space program again, please God, we will need it sometime.
Thank you.
Are there any more questions? All right, thank you very much, Dr. McCormack.